Welcome to Writing the Future of Cancer Research. I want to start by thanking Twist Biosciences for the opportunity to present today. My name is Ken Chahine. I'm CEO of Helio Health, and I'm going to share with you a little bit of our data and some of our plans in the future. So first, to introduce our team. As I mentioned, I'm Ken Chahine. I used to work at Ancestry DNA, was there for about 10 years, started actually Ancestry DNA. Before that, I was at Pfizer and was also CEO of a gene therapy company. RF Nakuda is our CFO. He spent the majority of his career at Amazon, ran worldwide transportation and also worldwide Prime. Samuel Tong is our CEO of Helio China. Has been in multinational companies and led China for QIAGEN, Becton Dickinson, and Abbott. And then Ben Euler is our Chief Operating Officer, and he was with me at Ancestry DNA and also Ancestry Health. On the technology leadership, we have Richard Van Etten, MD, PhD. He's a professor at the University of California, Irvine, serves as our Chief Medical Advisor. Dr. Wei Li, who's an endowed professor of bioinformatics at the University of California, Irvine, is our advisor and leads our strategy on the bioinformatics side. And then Mindy Nguyen at Stanford is our expert hepatologist. She's a very prominent AASLD member, which is the top organization that provides guidelines for liver screening, and is an advocate for liver screening and surveillance. So let me start off by just sharing an overview of the company. As we think about early cancer detection, I think there's little question that over the next 5 to 10 years, that will be the dominant technology for cancer screening. And it's going to have a profound impact on the way we treat cancer. The question is, how do we get from where we are today, let's say 5 or 10 years from now? Who are going to be the winners and losers in terms of companies that are going to be providing these services? And I think if we look back, a lot of the basic technology that's being used for early cancer detection is available to everyone, right? A lot of people are using methylation or proteins. NGS is obviously a major component of this. So what's going to differentiate, the companies today from the companies that will be successful in the next 10 years? And we're firm believers that ultimately this is an AI machine learning driven algorithmic product, right, or device. And so we believe that having more data is going to be critical to really separate yourself from the competition. And one of the advantages that we have at Helio Health is that we're a global company. We have full operations in China. And our strategy really is to leverage China to access large number of samples at a scale, a cost, and speed that frankly cannot be reproduced in the United States or other parts of the world. So we really believe strongly in using and leveraging China as the initial R&D hypothesis generating research, if you will, that then can be leveraged to the United States and other parts of the world. We've done some of that already. I'll share some of the data that we have in terms of how data translates from China to the United States. We're a blood-based technology. I think we're all believers that blood-based is ultimately the way to go. We're also believers in methylation. We were early in the methylation space. We have 9 patent families that have been licensed from the University of California of San Diego, going back to 2015 when methylation in early cancer detection was not as prevalent as it is today. We will, however, use proteins or other biomarkers that are available that potentially could provide improved performance. Liver cancer is our first indication, and we have 2 clinical studies, one that's been completed in China. We call it the Victory trial. It was almost 1,100 subjects, and we looked at liver cancer at various stages compared to liver disease, but no liver cancer such as cirrhosis. And what we found was that trial is fully complete, registration trial. What we found is at stage 1, we had a sensitivity of over 90%, so it performed quite well, and the specificity was greater than 95%. So we've already submitted the formal package to the NMPA, as it's called in China, for registration. We're the first company, to our knowledge, to have submitted any kind of registration for a blood-based test. So we're very pleased with that. Anticipate, a year through this process, registration process, and having approval by the fourth quarter of this year. We're also in parallel going through an FDA clinical trial also for liver. This is a prospective trial. I'll share a little bit about the trial design and some of the data in a few slides, but suffice it to say that we're seeing stage 1 sensitivity around 86%, so performing quite well, and specificity of 91%. And we'll talk about what the standard of care is in liver cancer and why these numbers are so compelling. So really where the company now is moving is from this clinical stage of the liver cancer into the market. In China, we've launched as an LDT and just ramping that up right now. Obviously, the FDA, the Chinese FDA or the NMPA will be a key milestone for us to accelerate sales. In the US, we do have breakthrough status, and so we are recruiting that trial. We anticipate the recruitment to be done at the end of the second quarter, and then from there we would, basically read out the data, move into an LDT strategy initially, and then ultimately a PMA and market authorization. While we're really focused on liver, we think it's a great indication. We are a platform technology. We're very interested in colon, breast, and lung cancer, and we'll be advancing those in 2022. You can see that we're working with world-class partners, not just here in the United States, but also in China. Sun Yat-sen University, for example, in Guangzhou is the largest cancer hospital in China, and the NCC is equivalent to our National Cancer Institute here in the United States, and has been an amazing partner in terms of samples and really providing sort of real-world screening opportunities that are difficult to come by otherwise. And then you can see, I'll share a little bit about the market opportunity. I think many of you, if you've looked at the early cancer detection, will agree that this is a great market. So super briefly here, just to talk about a great market, TAM is about $150 billion a year. That's assuming 4 cancers: liver, colon, breast, and lung for China and the United States. And one of the things that's incredibly appealing about the early cancer detection space is, unlike most genetic tests and certainly even most diagnostics, this is something that's a repeat business. In other words, if you come in and you're negative, you still have to come back the following year or 2 years later to get an additional test. And this repeat nature of the business actually makes it, from a commercial and financial standpoint, incredibly attractive. And so let me just spend a minute here on why liver cancer, because there's a lot of companies in the United States in particular going after colon cancer. Great market. Obviously, Exact Sciences has done a fabulous job demonstrating the opportunity in that market. And there's some other advantages to colon cancer, but we really love liver cancer for a few reasons. One, the market is large and growing. Because we're a global company, hepatitis B is one of the major risk factors for liver cancer, and China alone has 80 to 100 million people with hepatitis B, and actually represents half of all liver cancer in the world. In the United States, it's also a nice market. We have about 6 million people with liver disease today, and that doesn't even count non-alcoholic fatty liver disease and NASH, which I think we all know is an epidemic and growing. And sadly, because of that, liver cancer is one of the few cancers that's growing and growing pretty rapidly. Looking at a few articles, one in particular in the Lancet, showing growth of about 2 to 2.5% a year for liver cirrhosis and liver cancer. So it's a very nice market, and it continues to grow. The second thing is that the universal standard of care for ultrasound, for liver cancer, is ultrasound. It's every 6 months. And the last point is that ultrasound, unlike a colonoscopy, which is inconvenient and costly, an ultrasound's but performance is good. An ultrasound's performance is actually quite poor, and that is the sort of the standard that we have to beat. So super quickly, just to give you a sense of how we got to the product, we did a top-down marker selection. Started off with about 3 million potential markers. Continue to sort of streamline that through bioinformatics. At the end, we ended up with around 38 markers for this initial test. Roughly 27 of them are hypermethylated and 11 are hypomethylated. So that is really our methylation probe set. And one of the things that we really encouraged is we went back and then looked at where these methylation markers, sort of ended up. And what we found is that they correlate quite nicely and are annotated to many of the sort of promoter regions, as you would expect, to cancer genes, and several of them, and many of them actually, are linked to liver-specific genes that are associated with cancer. So obviously that gives us a lot of confidence in the selection of the markers. Lastly, we've applied a proprietary algorithm that was recently published in Nature Communications. We call it CHALM. It was developed by Dr. Wei Li. And basically what that looks at is not just looking at the sort of average methylation across all of the different DNA that you see in your cell, but actually trying to look at the methylation at individual cells. And what he has shown, and his lab have shown, is that by looking at how many different individual cells, if you will, are methylated versus trying to take an average, it becomes a much more powerful way of predicting whether that individual has cancer or not. And I don't have enough time to go through the data, but you can see the publication here, and that's something that we have filed a patent application on and are using in our technology. The trial design, super quickly for the US, is fairly standard for a screening trial or surveillance trial. A high-risk asymptomatic subject comes in, they get our helio liver test, they get an ultrasound, which is a standard of care, and also AFP, which is commonly used. The clinical truth is a multiphasic MRI with contrast. And then in liver cancer, what you find is something called the LIRAD score. A LIRAD is scored from 1 to 5. A 1, 2 is a negative, a 4, 5 is a positive. A LIRAD 3 is indeterminate, and those individuals come back for a second MRI in 6 months to see if that indeterminate has resolved. It can either obviously resolve as continuing to be negative as a 1, 2, 3, or has progressed to a LIRAD 4 or 5, in which case it was considered positive. According to the literature, about 20 or 30% of those LIRAD 3s will progress to liver cancer. So if we look at the data super quickly, we have continued to recruit subjects in this clinical trial. We've recruited, I believe, almost 400. In addition to our retrospective data set, we've used this data set to further optimize the algorithm and ultimately use that as a sort of independent training set to confirm that in the setting of the clinical trial and in the setting of real life clinical practice, right, the algorithm works as expected. And really briefly here, show you on the left, top left, the performance of ultrasound. The way the trial is designed, we have found that almost every patient that has been determined to have liver cancer has stage 1 cancer. We have very few, I believe only a handful of stage 2s, no stage 3 cancers and no stage 4. And so ultrasound, not surprisingly, is performing particularly poorly, even more poorly than we see in the literature. AFP, is performing pretty much as it's reported in the literature. And as you can see, our helio liver test, which is our methylation plus some additional protein, so it's a multi-analyte test, is actually performing quite well. And so on the right, you can see sort of the pie graph, if you will, of the performance of ultrasound. 30% sensitivity, 98% specific, and our helio liver test is performing at 86% sensitivity and 91% specificity. So we're very happy with these results. And the goal here is now that we're locking the algorithm, we're continuing to recruit subjects that are fully blinded by a third-party CRO, as you would normally expect, and then we will begin to start testing those samples with the locked algorithm probably in the second quarter once we've completed a lot of the recruitment that we need. Switching gears just a little bit, I said we had done a registration trial called Victory Trial in China. These are the hospitals on the left that were used. It was a prospective trial. In China, it's done slightly differently than the US. I don't have enough time to go into it right now, but it was these three hospitals. The total subjects, as I mentioned earlier, was almost 1,100. And as you can see, in China, it is a combination of ultrasound and AFP, which is not used not used that way in the United States. And a combination of ultrasound and AFP for this data set, you can see that it performed at 51% sensitivity and 99% specific. And our Helio CF DNA blood test at stage 1 had a performance of 92% sensitivity and 97% specificity. So that's a very good trial, and these are the data that were submitted for the pivotal registration trial. One of the things that's really important that I mentioned, right, that one of our key competitive advantages and what we believe can create some separation between us and the competition is the number of samples. And so I'm just showing you here super briefly an example of the samples that we're able to collect. So you can see that we have several hospitals we're working with right now that we can collect samples for any indication. And then with NCC, we're working on 3 different studies: liver, colon, and lung. Liver is just to continue to confirm and actually look at potential differences that may occur if the underlying disease that leads to the risk profile for liver cancer is, for example, hepatitis B, hepatitis C, alcoholic cirrhosis, non-alcoholic fatty liver. We're really interested in trying to understand further if there's any algorithmic optimizations that need to happen based on the underlying condition. For colon cancer, and as you can see, we've recruited 5,000 subjects already. It's continuing to actively recruit. 100% of those subjects have had a colonoscopy, and in many cases, pathology as well. So very eager to get started. We're going to be doing whole NGS sequencing on those and some discovery. And then lung cancer, which is fairly wide open, is something that we're very interested as well. We have over 7,000 samples that's actively recruiting. And again, all of those subjects have a low dose CT scan, which is a standard of care. So we are, fulfilling, if you will, our thesis that we can get a lot of samples in China. These samples were recruited very quickly. You can see the scale, and the cost of these are literally a fraction of what it would cost in the United States. So again, eager to get started on this. One of the questions that we get asked, and frankly we asked ourselves, right, is that's great that you can get a lot of samples in China. How much or how well does that data or those data translate into a U.S. population? And let me just show you just super briefly 2 slides on this. The first thing we did is, because we got NGS up and running first in the United States before we got it running in China, we basically used the retrospective data set that we had, used a lot of the climb samples that we had that were prospective, developed a first generation sort of algorithm. And then once we had that locked down, these are the 38 markers that we had talked about, we got an independent set of around 400 samples from China, completely naive, had never seen or been used in any way for any of the marker selection or any of the optimization. And we did 2 things. The first thing we said is, okay, how do these 38 markers look, and what's the concordance between what we're seeing in the US and in China? And I think just at a glance, you can kind of see that the concordance is pretty amazing. At the top, you're looking at both hyper and hypomethylated markers. At the bottom, you're looking at hypomethylated markers in China. The concordance is, about 95%. There's 1 or 2 markers that you can see are discordant, but other than that, we're seeing great concordance. So this was, really, really encouraging to us that while there are ethnic differences, it does seem like the underlying biology and the markers that were selected seem to translate quite well between the 2. And of course, when we did the performance, you can see the performance on the left on this one data set and the performance of the China data set. It's actually performing quite well. So we're super encouraged without any modifications whatsoever. We're seeing great performance. And as again, we certainly anticipate, and we already have some data, that we will have to tweak the algorithms by different geos, because the underlying condition that leads to the liver cancer risk are different. And it's our hypothesis right now, based on some data, that is probably the major differentiator that we're seeing, not so much the ethnic background of the individual, but we'll continue to look at that. Nonetheless, without any modifications, we're very, very pleased with the performance, and this really continues to lend credence to our hypothesis that we can use China as a discovery engine. So just to end up, as we look at early cancer detection, we think it's going to be, really important that we focus on a few things. One, we do firmly believe that data is going to win, and we're working with Twist on their methylome and a bunch of other technologies that they have to continue to advance that. So all those studies that I showed you earlier, we fully anticipate using sort of the Twist platform to do the discovery work on that. So we're really eager to get started. Second, we have to make it simple. A colonoscopy is not simple. An ultrasound is not simple. We know that the best way to beat cancer is to treat it, is to catch it early, but we certainly don't make it easy today with the technologies that we have. So a simple blood test is absolutely going to revolutionize this. But we would argue that even beyond that, we need things to make it simpler for the consumer. Increasing awareness. I mean, we know from colon cancer that awareness is way lower than it should be. For liver cancer and other cancers, it's really poor. And cirrhosis of the liver in particular can go silent for a long time. We need to get people to increase awareness, go to their doctors, get diagnosed, and then ultimately get into a screening or surveillance platform. Look, in-home testing is something that absolutely was already a major driver. COVID's continuing to drive that. And we need to figure out a way if we can to make this even simpler for the consumer, potentially even having an in-home test where you don't have to go to the doctor, wait in line, etc., to even get a blood draw. Maybe this can be done at home, and this is something that we're actively exploring. And from a business standpoint, we really do believe this should be a low price strategy. Shouldn't try to capture all your value at one time. And so our goal is to price this very aggressively, at least at par to ultrasound and potentially lower than ultrasound, because we think that it's super important that everyone gets screened. And when we make this simple, we don't want to make this a challenging decision for the reimburser. So anyway, I'll end there. I want to thank again Twist for the opportunity to present Helio Health, and we hope you enjoy the conference. Thank you.